expressions are lost. The partial restoration and maintenance of the deep B cell property is possible. For example, in this case, we developed the culture condition we call dermal papilla activation culture condition, or DPAC. This is a culture media containing the activators for the Wnt and the FGF and BMP signaling pathway. But you can partially find that in this case, alkyl phosphatase expression was maintained when treated with DPAC. But if you perform the hair reconstitution sandwich assay using the cell aggregates from the conventionally cultured human dermal papilla cells with DPAC restored dermal papilla cells, we can get some hair follicle-like structures, but they are actually not the hair follicle. So, the current major hurdles in human hair follicle regeneration is limitation in starting material and loss of intrinsic properties in vitro. So, are there any better material? This is a question that I myself had during my research aiming at the human hair follicle bioengineering. And actually, human iPS cells hold promise for skin research and bioengineering. Human iPSCs can be generated from keratinocytes, melanocytes, fibroblasts, and dermal papilla cells, and especially dermal papilla cells are very promising material for generating iPS cells because some of them are intrinsically express the Yamanaka reprogramming factor. So human DP cells can be induced into the reprogrammed into the human iPSCs just with one factors in case of mice. While still, to generate the human iPS cells from human cells, we need 4 Yamanaka factors. And human iPSCs, vice versa, can be induced into keratinocytes, melanocytes, and dermal cells. So, these are the major components for the hair, since these are the major components for the hair follicle regeneration. So hair follicle regeneration may be possible using the iPS cells. And interestingly, the iPS cell can be generated in a plucked hair follicle. So via reprogramming and the differentiation process. And as you know, that iPS cells are theoretically unlimitedly divide and proliferate and differentiate in multiple lineages. So maybe plucked hair follicle via human iPSCs with those regenerated cell populations can provide the nice material for the human hair follicle bioengineering. So let us think about the advantage of human iPSCs for human hair follicle regeneration. One is obviously unlimited supply. Second, multipotency, and less ethical issues. But in my talk, I really would like to focus on the possible adjustability of differentiation or differentiation of different. Sorry, maybe I should rephrase this one to adjustment, but the adjustment of differentiation status, and also a single material for both epithelial and mesenchymal components. So, in conventional approach that we have been using, we isolate both the epithelial stem cells and the inductive mesenchyme and expand those cells to a certain extent. But meanwhile, the number of the cells with intrinsic property will be decreased, and use those small number of cells or impaired with possibly impaired intrinsic property to regenerate incomplete hair follicle structure. These are the results in my laboratory, so maybe we have no good terms, but in case of iPS cells, this can be possible. So iPS cells induce those cells to precursors for both epithelial and and mesenchymal, and educate those populations to be a stem cells or precursors, early precursors, which has the very strong morphogenic capacity. And these populations should enable the regeneration of hair follicle structures. So let's start discussing from the epithelial cell components. By adapting the retinoic acid and BMP signaling, we are able to use the keratinocyte precursor cell or epithelial precursor cell, we call EPCs or KPCs, from the iPS cells. We generated iPSCs from the iPSC KCPs from the 3 independent cell lines, but as you can see, those iPS cell lines can be converted to the early keratinocyte progenitors expressing K18 and K14, as you can see with a green staining here. Interestingly, when those generated iPS-derived keratinocyte epithelial precursors are co-cultured with the human dermal papilla cells, and gene expression level of those cell population are compared with human adult keratinocytes, iPS-derived cells expressed similar or even stronger level of hair-related gene expression than normal hair follicle keratinocytes. And also, the human-induced iPS cell-derived cells can stimulate the DP biomarker expression in co-culture system, as indicated by black light here, suggesting that the human iPS-derived keratinocyte precursors more efficiently communicate with the dermal papilla cells in vitro than human adult normal keratinocytes. So next, we picked up the cell lines that most efficiently responded to the dermal signal and generated the human iPS cell-derived epithelial cells and combine them with mouse neonatal dermal fibroblasts and perform the patch assay. Interestingly, after 2 or 3 weeks, we can observe the hair follicle structure like this, and those hair follicle structure express the human specific cell markers, and as you can see, express the human bulge markers or K14 gene expression. This is a positive control. This is a hybrid hair follicle that's obtained from iPSC, suggesting that at least partially human iPS cell-derived epithelial components can contribute to the hair follicle structure in vivo. And recently, the group from the University of Pennsylvania successfully generated the bulge stem cells from the human iPSCs. And what they did is that they further modified the conventional or previously published protocol using the retinoic acid and BMP4, and they combined EGF and regenerated the human bulge cells, as demonstrated by the high expression of the CD200, as I mentioned before, and the keratin 15 established marker for the human bulge cells, and you can get successful human type hair reconstitution. So maybe you are encouraged to use this protocol, not ours, for human hair follicle generation of the human hair follicle epithelial components at least. So, preparation of the human iPSC-derived follicogenic stem cell was completed, seemingly completed. Next question would be, how can we regenerate a trigogenic mesenchymal cell from human iPSCs? And what is a protocol that's enabling us to generate the human iPSCs, trigogenic dermal papilla cells from human iPSCs? The work by my colleagues demonstrated that at least partially, the dermal papilla cells derived from the neural growth cells. So we are taking advantage of this fact for regeneration of the human iPSCs. So human iPSCs, there are two approaches conceivable. One is via mesenchymal stem cell and using retinoic acid to generate the human neural growth cell and generate the DPs. Or you can directly convert the iPS cells to the neural growth cell to the DPs. But until very recently, there are no established protocol to stably generate the human neural growth cells. So we first started to generate the human mesenchymal stem cells from the human iPSCs. So human iPSCs, embryo bodies are formed, and then using the mesenchymal induction condition, and we can get the human MSCs, as you can see here. These are the process, and the human MSCs, they can be passaged. And these are bone marrow derived human MSCs as a control comparison. The generated human MSCs express the human mesenchymal markers as indicated by here. These are the positive control, but not the bone marrow lineage markers. And importantly, those components can be differentiated into osteocytes, adipocytes, and the chondrocytes, demonstrating that they are mesenchymal stem cells. The problem with the mesenchymal stem cell is that the mesenchymal stem cells are highly heterogeneous cell population. They are defined by function. So that's sometimes called a problem. So we'd like to focus on the really pure and highly pluripotent and multipotent mesenchymal stem cell population. And recently, our colleagues reported that the subpopulation CD271 positive, CD90 positive human mesenchymal stem cells are small, quiescent, but highly multipotent subpopulation of mesenchymal stem cells. But the problem with those cell population was, as you can see here, they account less than 0.1% of the bone marrow derived mesenchymal stem cells. So it's extremely hard to use those cell population for the downstream analysis, including tissue regeneration assay. As you can see here, the percentage is very low in this sample. But very interestingly, the mesenchymal stem cells regenerated from human iPSCs with our protocol, as you can see here, in this sample, we can see up to 20% of the mesenchymal cells are CD271 and CD90 positive. And if you induce those cell population into the three dermal lineages, they successfully differentiated in multiple lineages, suggesting that they are indeed the potent mesenchymal stem cell subsets. So the next question would be, these CD271 and 90 double positive cells can be endowed with some DP properties. And as you can see here, those double positive cells are exposed to the DP induction condition. And interestingly, this is a positive control, and in case this is an alkyl phosphatase thing, alkyl phosphatase expression was significantly upgraded after those cell population is exposed to the DP induction condition. And interestingly, please pay attention to the morphology of those cells. Some of the cells are cuboidal, and they are similar to the human dermal papilla cells, functionally restored in the DPAP condition that we described, that I mentioned before. And as you can see, these are the mesenchymal-derived human dermal papilla substitute cells that we call them. And these are the dermal papilla cells in culture, but some markers are more highly upregulated in the human iPS-derived dermal papilla substitute cells. So, and we go on to the functional assessment of those cell populations. And what we did is we co-culture the human dermal papilla cells or human DPSCs, iPS-derived DPSCs with human keratinocytes, and assess whether those cell populations can communicate or reproduce epithelial mesenchymal interaction in vitro or not. As you can see here, the keratinocytes upregulated hair follicle-related genes equivalent to the cultured dermal papilla cells when they are co-cultured with the human dermal papilla substitute cells. And conversely, the DP biomarker expression was also similarly upregulated in cultured dermal papilla cells and the human dermal iPS-derived iPSCs. And currently, the further characterization of those cell population is in progress, and we are really expecting to package this story in very, very shortly. Let me quickly explain you about the approach to the regeneration of human iPSCs using regeneration of human iPSC-derived neural crystals. As I mentioned before, the protocol has not been stable, but recently the group in Stanford established a protocol to stably regenerate the neural crystals from the human iPSCs. In case of here, you can see that those cell population induced from the human iPS cells efficiently express the CD271 and CD59 cells, as you can see here, which is a marker for the neural crystals. And the question would be, deep induction is possible by the condition that we establish for the mesenchymal stem cells. But at least until now, the viability of those cells in our condition for the MSCs are not that great. So maybe I should put question mark in here. So in conclusions, we can now generate the human iPSC-derived KC lineage for hair follicle bioengineering, and it's possibly we can regenerate the mesenchymal components also from the human iPSCs. So human iPSC can be useful for dissecting hair follicle biology, and may provide promising material for human hair follicle bioengineering. At the end, I'd like to thank for all my mentors and collaborators and all the lab members in my hair lab in Keio University Hospital. Well, thank you very much for your attention, and 감사합니다.
Thank you very much for your nice informative lecture. Now this lecture is open for discussion. Please do any questions or comments. No question? Yes, please.
Excellent talk. I'm wondering about the mesenchymal components when you use the markers for BMP4, WNT. A lot of the alkaline phosphatase, a lot of those are found in bone, right?
Yeah.
So how do you know that it's really a DP signature, not
Oh,
bone?
yeah, that's a very good question. And and currently we are generating the microarrays to see to compare the global gene expression within those cell components. So that should answer your question.
Other question? Yes, please.
Ohyama, I really enjoyed your talk.
Thank you very much.
Do you think the origin of the iPS cell would affect the inducibility for the hair follicle generation?
I think that's a very good question. And for example, in case of epithelial cells, they are very different. I mean, in terms of the efficiency in the upregulation of the hair follicle markers. So perhaps the origin can affect the intrinsic property of the iPS cells. So, but it's really hard to compare the multiple cell components. So the iPS cell lines that we are using is like our handmade is a very conventional iPS generation approach from the scalp dermal fibroblasts. Yeah. But I think your question is excellent.
Okay, another question? Yes, please.
I have a question regarding the hair follicle markers. So you decided to take keratin 75, the marker for hair follicle. And keratin 75 is not the major keratin expressed by I think it's cuticle of thyroid.
Yeah. I mean, this is, I think, a very, very quick checkout. And the reason why we picked up is like we use those markers for the previous experimentation. So that's the reason why. And I think your point is correct.
Okay, last question. One more. No? Professor Ohyama, could you please tell me the next step of your study very briefly?
I think there are two aspects, and I have discussed with some of the audience yesterday, but I think there are two aspects in the hair follicle bioengineering. The one aspect is how we apply those techniques in the clinical setting. And for the clinical setting, maybe it's much better to use the hair follicle-derived stem cell components or dermal components. But at the same time, we need to show the cutting-edge technology. Or in other words, hey everybody, we can do this. And I think that what I am doing, at least with the human iPS cell, is that that's within that category. And the next stage will be, I'd like to combine the human-derived epithelial components from the iPS cells. And our group have been successfully demonstrated that melanocytes can be regenerated from the iPS cells. So I really would like to hope the pigmented hair follicle only composed of the iPS-derived cell components.
Okay, I'd like to close this lecture. Thank you again.
Thank you very much for all audience. Thank you very much.